Hello guys, as we have already discussed about the GIT tract which includes salivary secretion and gastric secretions. So today we are going to discuss about the pancreatic secretions. It is also related to the previous lectures which we end with gastric secretion. So pancreatic secretions, that secretion from the pancreas which is exocrine, that will be considered as pancreatic secretion in GIT. So pancreatic secretion, it is always alkaline, having pH of 8.3. Pancreatic secretion is rich in bicarbonate ion. It is rich in bicarbonate ion. So in food we have carbohydrates, we have lipids, and we have proteins. So for carbohydrates in the pancreatic secretion, we have pancreatic amylase, which do digestion of carbohydrates. In pancreatic secretion, for lipids, we have pancreatic lipase, and in pancreatic secretion for proteins we have three enzymes. Number one, trypsinogen. Number two, we have chemotrypsinogen. And number third, we have procarboxypeptidases. So those enzymes which end with gin, they are considered as inactive. So all the protein enzymes that are released from the pancreas, they are in inactive form. That is why these enzymes are called zymogens. So they are activated inside the intestine. Why pancreas secretes these enzymes in inactive form? As we know that protein enzymes are so powerful that if they are released from the pancreas in active form, they will eat out the pancreas. So in order to prevent the autodigestion of pancreas, these enzymes release in inactive form from the pancreas. Now how they are converted? Like suppose in small intestine, the trypsinogen, it is converted into trypsin. And we know that trypsinogen is inactive form. And this is active form. These are converted by an enzyme which is called enterokinase. This is present inside the small intestine, and this do the conversion of trypsinogen to trypsin. Now how these pancreatic secretions are regulated? So now we will discuss the regulation of pancreatic secretion. The regulation is totally under the control of two hormones. Number one is cholecystokinin P2, and number second is secretin. Now what cholecystokinin do? It does the contraction of gallbladder. gallbladder. When gallbladder is contracted, what will happen? It will release bile, and this bile help in digestion. Now, what this P2 do? This is called pancreazyme. P2 is called pancreazyme. What it do? This is a pancreatic secretion which is rich in enzyme, which is rich in enzymes, and it helps in the digestion of the food. Now secretin, we come to a secretin. So it is also a pancreatic secretion, but this is poor in digestion. It means that those secretion from the pancreas which is regulated by the secretin, they are poor in digestions because they do not have enzymes, but this secretion is rich in bicarbonate ions. So because of secretin, pancreatic secretion is alkaline. Because of secretin, most of the pancreatic secretion is alkaline. So what it does? It actually do the acid neutralization. It do the acid neutralization of the small intestine. When the acid come from the stomach, when they come from the stomach, so to prevent the damage of the small intestine from this stomach acid, the secretin secretes bicarbonates from the pancreas. Now we will discuss about the bile, that what is bile and what are the functions of bile. It is produced by liver. Almost 500 milliliter per day bile is produced. It is stored in gallbladder. Bile is stored in gallbladder, and in the process of storing in the liver, concentration of bile occur. So here two important points which should be noted, that number first, that liver bile, it is 97% consist of water, and that bile which is produced inside the liver and that reach to the gallbladder where the concentration take place. So gallbladder bile, it consists of 87% of water contents. The liver bile, it is 97% contents of water, and the gallbladder, it is 87% of water contents because of the concentration of the bile inside the gallbladder. Now we will discuss about the constituents that bile consists of. Constituents of bile, that bile consists of which things? So it consists of bile acid, it consists of bile acid, it consists of bile salt, it also consists of bile pigments, and it also consists of bile cholesterol. These are the constituents of bile. Now we will discuss a little bit about bile acid, that what type of acids are present inside the bile. So there are two types. One is primary bile acid and the other one is the secondary bile acid. So the primary bile acids include cholic acid, and it also include condeoxycholic acid. it also consists of this. So these two are considered as primary bile acids. When these two acids, they reach to small intestine where any bacteria act on these acids, they convert into a secondary acids, bile acids. So secondary bile acid are deoxycholic acid. It is deoxycholic acid, number first, and number second is lithocholic acid. Number two is lithocholic acid. So these two acids are produced inside the intestine when any type of the bacteria act on the primary bile acids. So these are converted into a secondary bile acids. Now we will discuss a little bit that what are the important bile salts are present. So bile salt are almost, it is salt of the sodium or potassium. It is a salt of sodium or potassium. So for example, attached with the bile acids, for example sodium glycolic acid, sodium glycolic acid. This is the salt which is present inside the bile secretion from the gall bladder. Now we will discuss a little bit about the importance of the bile acids. Importance of bile acid. Number one, they undergo conjugation process in liver. They undergo conjugation process in liver. Number second, during conjugation, bile salt combined with glycine, bile acid combined with glycine or taurine. So these two are the importance of the bile salt that in the liver, it due to bile acid, the conjugation process takes place and it combined with glycine or taurine. So now we will discuss a little bit about the function of the bile, that what is the function of bile. So number one, the most important function is it helps in digestion and absorption of the fats. It helps in digestion and absorption of the fats. Number second, it also helps in excretion. These are the two important function of the bile. So now we will discuss a little bit about the regulation of the bile, that how bile is regulated. So there is a one word you should also remember, that what does it mean? Choleratex. What is choleratex? So choleratex is any substance that increase bile secretion, so any substance which is related to increase the bile secretions, we call that substance as choleratex. For example, what are choleretics? For example, so the most potent choleretics is bile salt itself. Bile salt itself regulates the increase or decrease of the bile productions. Number second, secretin. As we all know that secretin, it also regulates bile secretion. Number three, parasympathetic division. Parasympathetic means autonomic nervous system also regulate the productions and secretion of the bile from the gall bladder. Number second, that release of the bile. What cause release of bile from gall bladder. So the release of bile from the gallbladder, it takes place when the gallbladder is contracted. When the gallbladder is contracted. What we have already discussed, that how gallbladder is contracted. Contraction of gallbladder, it takes place by cholecystokinin enzyme. Cholecystokinin enzyme, it do the contraction of the gallbladder. When the gallbladder is contracted, so it causes the release of the bile from the gallbladder. Now for MCQ's you should remember that any substance that cause contraction of gallbladder, any substance that cause contraction of gallbladder, that substance is called cholegogies. That substance is called cholegogies. And we also know that cholecystokinin cause the contraction of the gallbladder. So cholecystokinin is one of the important cholegogies we have discussed to cause the release of the bile from the gallbladder. So till now we have discussed pancreatic secretions. With pancreatic secretions we have discussed bile, its regulation, functions, and how it regulates. Now the last point towards the GIT secretions we have to discuss here is the intestinal secretion. Intestinal secretion, that what are the secretions that take place inside the intestine during GIT tract. So the place we called it as a crypts of Lebo-Kahan. Crypts, you should remember that this place, that crypts of Lebo-Kahan. This is the place inside the intestine which contain many intestinal cells, and those cells are responsible to release the intestinal secretion. This is the place which is responsible for the secretion of intestinal secretions, which include many cells, and those cells secretes intestinal secretions. Among them, number first cells, we have a surface or goblet cell. We have surface or goblet cells. What these cells do? They secretes mucus, and this mucus is rich in bicarbonate ion. So any acidity that come from the stomach, this mucus, it prevents this intestine from the acidity of that. So it does the acid neutralization of the intestine. Number second cell among them is intestinal epithelial cell. Intestinal epithelial cells. So intestinal epithelium or intestinal secretion, it gives a special name which is called as succus entericus, which is called succus, this space is called succus entericus. Succus entericus. And this is the place where these epithelial cells secretes their secretions. In this space, these cells secrete their secretions. Number third and the last cells that cause the intestinal secretions are Penit cell. These are Penit cells. They are present inside the intestine. So they are located at the base. Penit cells located at the base. They have high level of zinc. And they also produce defensins. And lysozyme. They also produce defensins and lysozymes, which are antimicrobial agents. agents. So these also act as a immune system for the intestines, that they secrete defensins and lysozymes. So till now we have discussed the whole secretions that that is responsible for GIT tract. So if you are new on our channel, please subscribe our channel. Thank you.